We are down to it. Good morning. It is Monday, May 4th. We are reaching the end of this class, rapidly approaching the end. We are on to our last section, the reproductive system. Another quick 2-week turnaround on this. Only really one assignment. Our unit review, unit 27, is going to be due on 1 week from today, May the 11th. I do have a 2-point extra credit survey set up for you based on the number of technical issues that we had. We had about a similar number of technical issues for the this most recent lab exam as the previous one. So I'm trying to decide what's best for students, and so I want your personal opinion of that. I know what I have a preference for, but I'm willing to respect your preferences as well. Please complete that survey. I know a couple of you have already. I would like more of you to do that to help me make an educated decision on what you think the best preference of your format for your lab exam is, and that's due by 10 p.m. tonight. Our next lab and lecture exam is going to be on Wednesday the 13th. Again, the format for the lab exam is going to be determined based a lot on what you guys say and then what is finally determined on that, but the lecture exam is the lecture exam. And I'm still working on the final exam format. The good news is got all of your stuff graded this weekend, so I was very busy doing that, but the downside of that is I didn't have any opportunity to look at and work on our final yet. So I don't know what the format for our final is going to be. Speaking of the exams, the scores were decent, better than the last one, which is expected. The digestive system is always a little bit of a nice palate cleanser, pun intended, because it's a simple, straightforward, single unit, single organ system. So they tend to be a little bit better. The lecture exam average was a 47.6, which is a little over 73%, and our lecture exam was just over 72%. But as we keep talking about, the only grade that matters is the overall grade. And so far, everybody is hopefully doing as they'd like to be doing, but the class as a whole is doing pretty decent, with the overall grade hovering right around 81%, which is not a bad place, although certainly there are some people who have some room for improvement on that. After lecture today, because again, if I do it before a lecture, then you guys are all looking at your exams and not paying any attention to me. I will be releasing your exams and the grades today after class. Obviously, the grades you'll be able to see from now till the end of the semester, but the exams will only be available for the next 24 hours. So again, just like last time, you'll have 24 hours to review it. If you have any questions, I tried to put a couple more comments into the grades that I've provided for you guys to answer any questions that way. But if there's anything that's unclear, please email me. We can either meet or we can correspond by email to go over your exams. And then tomorrow when I start my afternoon class, I will be locking the exams again. So you'll have 24 hours to take a look at them. All righty, questions on any of that? Excellent, we have a few more stragglers who have worked their way in. Glad to hear that, glad to see that. All right. Where's my chat window? I realized I'm asking for questions. I don't have my chat window up. There we go. Perfect. All right. So it looks like that is pretty simple and straightforward. So excellent. Let's go ahead and switch gears then. Hide my free face and switch to the lecture. All righty. That goes there. And bring that up. Perfect. Sorry, takes me a moment to get everything set up. All right, so we are on to our reproductive system. The reproductive system is unique in a couple of ways. Of all of our organ systems, it is the one, excuse me, long weekend. It is the one organ system that is not continuously functioning throughout our entire life. Obviously, your cardiovascular system doesn't wait 13 years before it becomes active. That would be a problem, right? Your respiratory system doesn't take Sundays off or anything along those lines. Most of our organ systems, when we talk about them, they're continuously functioning organ systems. But the reproductive is not the case. It is not a continuously functioning. It starts and it stops, and it starts and it stops again for females. For males, it is a prolonged delay before it begins and then is continuous after that. So it is very different in its system that way. The other big difference with the reproductive system than our other systems is this is the first time we have meaningful sex differences. Right? With most of our organ systems, the cardiovascular system, the integumentary system, all those types of systems, there may be minor differences, between males and females, for instance, in the size of the heart or the amount of blood that is pumped or the heights or things along those lines. But those things aren't really meaningful. And you could even argue the urinary system, there are some distinct anatomical differences there, but both of those serve the exact same function. So while they are cosmetic differences, it really isn't difference in its functioning. The reproductive system is the one system where there are dynamic differences, significant differences, both functionally and structurally, between males and females. Of course, like always, we want to talk about the primary organs and the accessory structures like we normally do. And while, again, this is a reproductive system, it's always fun to say things like penis and vagina. When we talk about the primary sex organs, the penis and the vagina are not what we are talking about. What we are talking about are the gonads. The gonads are the testes in males and the ovaries in females. As we talked about, these are also endocrine glands, so they serve an endocrine function, but their job is to produce not only the hormones, but their job is also to produce the gametes. Right, the puzzle pieces, as we explained it to my daughter when she was young, that mom and dad put together to produce her. And of course, those gametes are the ovum in females and the spermatozoum, or spermatozoon, let's go continuous to singular, in males. Or ova and spermatozoa if we want to go plural for those. So our goal of the functions for the reproductive system are to have those gonads produce those gametes, those ova and the spermatozoa. And there we have them in the plural of the two forms there. But again, one of the big dramatic differences is females get the honor and the privilege of housing those puzzle pieces and developing them into a basketball-sized person over the series of 40 weeks, and then expulsing that person from their body. So they have to house and nourish and develop that offspring as well. However, as we also said, our gonads also produce our sex hormones. In males, that is the class of hormones known as the androgens, of which testosterone is the most common. And in females, of course, those are the estrogens, primarily estradiol, and progestins, primarily progesterone. The function of those hormones are to grow and develop many of the organs and tissues, right? When we talk about the change in organization, the change in development, being able to distinguish a boy from a man, a girl from a woman, right? Those are those type of sexual maturity characteristics that we are talking about, that we talked about during the endocrine system: secondary hair, enlargements of the adipose, enlargements of the muscle and connective tissues, and things along those lines. Obviously, those hormones are going to develop and mature the reproductive organs so that we can put those puzzle pieces together and propagate our species. And then we also, when we especially talked about androgens, talked about the roles they play in things like aggression or sex drive or things along those lines as well. Now, as I mentioned, I think we talked about this before, our goal is to understand the reproductive system the same way your goal might be to understand how an engine in a car works. Every car has an engine, but if you didn't really understand how an engine works and you had choices, then again, if you were trying to choose between studying the engine of a 1968 Volkswagen Bug and a 2021 Lexus, you'd probably want to start with the Volkswagen Bug. All right, it is far simpler. Both of them have engines that do the exact same thing, essentially. But the Lexus has a lot more bells and whistles, a lot more complicated components to it than that 1968 Volkswagen Bug. And so that's what we're going to do here as well. We are going to study the bug first, and then once we learn the bug, we can talk about the Lexus. And the bug of the reproductive system is definitely the male. The male reproductive system is by far the least complicated of the two. So we'll start here and talk about the basic components of this one, and then from there we will move forward. Again, as we talked about, we have our primary organs, and those are the testes in the male. And then essentially we have a duct system for transportation. And one of the important concepts of this, and again, one of the things that is very different in females, is in males, this is a closed duct system. Wow, that's not anywhere near how you spell closed. That was closed. Okay, excellent. It is a closed duct system. What that means is that the spermatozoa, when they're produced in the testis, basically have one place and only one place they can go. And that is true through the entire ductwork system of the males until they are expulsed out the end of the penis. This is not true for females. The ductwork system in the females is actually an open ductwork system, which means that the female reproductive tract is actually open to the abdominal pelvic cavity. One of the implications of this is involving sexually transmitted diseases. In males, obviously, in anybody, a sexually transmitted disease is a bad thing. But in a male, that sexually transmitted disease typically stays within the reproductive organs. In a female, though, because their ductwork is actually open to the abdominal pelvic cavity, those sexually transmitted diseases have the abilities to affect, much more likely to affect, other organ systems, like the digestive system or other organs that are in your abdominal pelvic cavity. So they're much more serious and much more concerning for females than they are for males because of the matter of that. Now, this closed ductwork system is made up of several structures, including the epididymis, the Ductus Deferens, the ejaculatory duct, and the urethra, as well as some additional structures inside the testes that we will talk about as well. Of course, there are accessory organs. Again, the puzzle pieces, the spermatozoa that are produced are not expulsed from the penis, the reproductive system, just by themselves. They need to be protected, they need to be nourished, they need to be maintained. And so semen is not just sperm, but also other components as well. And those components primarily come from these three sets of glands. We have two, although the illustration here only shows one of it, hiding here behind the bladder on this side, and there's one on the other side as well, are the paired seminal vesicles, or what are also sometimes known as the seminal glands. We have a single prostate, and then two paired bulbourethral glands, which are also sometimes referred to as Cowper glands. And then, of course, there are the external genitalia, the scrotum, whose job is to contain and protect or support at least the testes, and the copulatory organ, that is the penis. And here we see it from kind of a, I don't know what you would call this, transverse, partial transverse anterior view. And here we see the same thing from a sagittal section as well. Let's start first by talking about the scrotum. One of the concepts we will need to discuss, and a term we are going to be responsible for as we move forward, is the idea of homologous structures. Homologous structures are basically defined as different structures in males and females, but they develop from the same set of cells. So basically, what we have is a cluster of cells that in females serve one function and in males they serve another. And the first example of that we're going to talk about is the scrotal sac. The scrotal sac for the males comes from the same set of cells that in females form the labia majora, which is the outer layer of labia or lips that form a region called the vestibule that help to border and define the female reproductive external genitalia. In males, basically those labia extend and descend and then fuse together at the center to form the structure that is the scrotum that is going to surround and support the testes. There are many biologists that believe that our sole function as an organism is to reproduce, produce offspring, propagate the species. And if that is our primary function, then it's a little bit intimidating to think that the structures that are responsible for that, the testes, are housed outside the abdominal pelvic cavity. That isn't necessarily a very efficient way of doing things, right? It's a very dangerous and unprotected location, as anyone who has ever won $10,000 on America's Funniest Home Video has learned. However, the reason, as I'm sure you guys know, is to help to regulate temperature of the testes. As it turns out, the testes are not capable of producing viable sperm. And again, by viable, I mean capable of fertilizing the egg and producing an offspring if the temperature is too high. So by having the testes located in the scrotum outside of the abdominal pelvic cavity, we can keep the temperature lower than core body temperature and modify temperature to help to maintain efficient and effective gamete production. So again, the scrotum contain the paired testicles. The testes are about 1 inch by 2 inch in size. As I mentioned, the scrotum, and I will inefficiently draw this here. As I mentioned, the scrotum basically is a homologous structure for the labia majora, the females, where basically the cells extend out and project down to form that pouch-like structure. One of the ways that you can see this developmental change that takes place, and I'll just draw it as an enlarged line right here, is there is actually a septum. Let's switch to highlighter here. Down the center of the scrotal sac, there is a midline septum known as a raphe. For those of you who have scrotal sacs, I would encourage you next time you have the opportunity, heck, if you're not on camera, feel free to do it now. Look at your scrotal sac and identify that raphe that runs down the midline. If you do not happen to own one, then feel free to borrow one from someone else. Hopefully you're quarantined with someone of appropriate age and willing to consent to allow you to perceive the raphe. Otherwise, I'm sure there are plenty of opportunities on things like OnlyFans or other places like that, where you can ask individual males to expose their raphe to you, and I'm sure they would be happy to do that. Just DM them on Instagram, and I'm sure you'll get all the pictures of raphes you could ever possibly want. As I mentioned, the function of the scrotum is to help to regulate body temperature, and not body temperature, testicular temperature, to produce viable sperm. One of the interesting things about the testes is that, like the ovaries, they actually develop in the abdominal pelvic cavity. But at about the time of birth, they actually descend into the scrotal sac. Usually around the 7th month of development. However, in some instances, one or both testes do not descend into the scrotal sac. And since the testes are something that isn't required till puberty is reached, in most cases, the doctors would do nothing about them. They figure they'd wait a year or two to see if it would descend on its own. And if a couple years it hasn't descended, then they might go in surgery to induce the testis out into the scrotal sac. However, The policy on this has changed, and one of the main reasons for that is that the lack of dissension of the testes have been associated with an increased likelihood in testicular cancer. So if the testis stays in the abdominal pelvic cavity for a prolonged period of time, it dramatically increases the likelihood of that individual having testicular cancer. So now, typically after a month or so, if the baby's testes, one or both, have not descended, then typically they'll do the surgery to manually descend them into the testes at a much earlier age to avoid that increased risk of testicular cancer. When we look inside the scrotum, it isn't just a paper bag where you've got a couple of eggs rolling around inside. We need to have some protection. That protection starts, not surprisingly, especially when you think of the fact that the testes start in the abdominal pelvic cavity, with a serous membrane that wraps around them. And like most serous membranes, it has a double layer. Double layer of our serosa, a visceral and a parietal. And in this case, that serous membrane is known as the tunica vaginalis. Again, it is an extension of the peritoneum from inside the abdominal pelvic cavity. And as we can see it here, switch to this view, we can see that there forms a pouch-like structure around the outer part with the parietal, and then there is the shrink-wrapped visceral peritoneum on the outer surface of the testis itself. The testis is responsible for producing the sperm, but that sperm has to be released as part of the ejaculate through the penis. So that sperm has to be then sent back into the abdominal pelvic cavity via structures known as the Ductus Deferens or the vas deferens. Producing sperm is an incredibly metabolically active process. So we need blood vessels, both arteries going to and veins coming from the testes for all of that metabolic activity. And obviously it is a well innervated structure to provide that protection. And like I said, to provide that $10,000 on America's Funniest Home Video. So there are a lot of structures that need to go to and from the testis, even though it is outside the body. This is accomplished, as we can see here, by a thick fibrous connective tissue tube that connects and surrounds the testis, known as the spermatocord. Notice the spermatocord goes through that inguinal ligament we've talked about. We talked about this a little bit in the digestive system. That inguinal canal is an opening through the inguinal ligament. Both males and females have them. But remember, as we talked about in females, that inguinal canal is a potential space, right? The layer is closed and surrounded and nothing passes through it. Whereas here in the males, the spermatocord passes through that inguinal canal. And remember, we talked about how this in males, because there is actually a structure that passes through this, it is a weak point where, again, a hernia can occur. If there's too much pressure in the abdominal pelvic cavity, fluid, or some connective tissues can sometimes push their way out, bulging out through this weak spot, producing that inguinal hernia. All right. The other thing that we can see here, starting from the beginning, is that there are some specialized structures that are going to help to maintain that intrascrotal temperature. Again, the testes must be kept at a temperature about 3 degrees Celsius lower than core body temperature to produce viable sperm. It can produce sperm at a warmer temperature, but they won't be viable. And again, remember, I keep using that term. Viable means to be able to fertilize an egg, right? That ovum. If it's not able to fertilize that egg, then it's pointless. It's not viable, and it serves no function whatsoever. All right. There are three main structures that help to maintain the temperature. Well, I guess four. The scrotum itself obviously keeps it outside the abdominal pelvic cavity. But just because you're outside of the abdominal pelvic cavity, you are able to maintain temperature, but you can't control the temperature. You're going to be smaller than core body temperature, but we need to be able to maintain it more than that. And so basically there are three structures that help to assist in this. The first of these is actually, and let's switch first and draw, here's my scrotal sac. And we'll just draw one testis in here. Hold on, I want to undo that because I need a little bit more playroom. Here's our scrotal sac, and there is our testis inside of it. The first structure that helps us to maintain intrascrotal temperature is a layer of muscle that lines our scrotal sac. This layer of muscle that lines our scrotal sac is going to be smooth muscle. Right? So obviously this is something that is involuntarily controlled. And its function is to, well, if you look at it, it kind of looks like a curved muscle, kind of like the diaphragm is a curved muscle. And when a curved muscle contracts, the muscle gets shorter and the curve flattens out. By doing that, if you think about it, what this can potentially do is actually change the shape and size of the scrotal sac. And this can bring the testes closer to the core of the body to help to keep them warm, or the muscle can relax and it can descend, allowing for more space, allowing the testes to get further away from the body to lower the temperature. So by changing the shape of the scrotal sac, we are able to change the location, the relative location of the testis to the core of the body. However, there is an additional muscle layer Nope, didn't want to do that. Change that back to black. Drawing, there we go, light pink. There is an additional layer of muscle contained within that spermatocord, and this additional layer of muscle wraps around the testis itself. This particular layer is, oops, let's undo that. I'll write this off to the side, is actually skeletal muscle. And skeletal muscle, of course, can be voluntarily controlled. Now, again, that doesn't mean that a male is consciously aware of, hey, my testis is a little bit warm, maybe I should lower it inside of the scrotal sac, or hey, my testis is a little closed, maybe I should bring that up. It is skeletal muscle, but of course it is controlled by reflexes. The same way that, for instance, the skeletal muscle in your eyelid is controlled by reflexes. I know now that we are all at home, most of us have a lot less to worry about during the course of the day, but most of us aren't sitting there going, hey, it's been 3 seconds since the last time I blinked. Maybe I should blink now to wet and moisten my eyes. Actually, my left eye is a little bit drier than my right, so maybe I'll just blink the left side. All right, now I got to blink the right side again, or maybe I should just blink both so it doesn't look like I'm winking back and forth. Right? Clearly, you have the ability to close your eyelids and open your eyelids voluntarily, right? But that blinking reflex isn't something you have to think about any more than breathing is something. All right, now's the time when I have to inhale, now's the time where I have to exhale. So again, remember, just because it can be voluntarily controlled as skeletal muscle doesn't mean that that's our job. So usually it is reflexes that are controlling these things. And so these obviously can elevate and depress the testis in the scrotal sac. So notice there are two sets of muscles here that are very different from each other in their location, in their composition, and but their function is the same, to maintain that interscrotal temperature. So let's clear the drawing, and then we can see the pretty words that go along with this, and we can identify those two sets of muscles. The smooth muscle within the scrotal wall is what is known as the dartus muscle. And the skeletal muscle that wraps around the testis is the cremaster muscle. And again, skeletal muscle versus smooth muscle, involuntary control versus voluntary control. While you're asking that male-persuaded individual to expose their raphe to you, you can also ask them to flex their cremaster muscle for you as well. And you can see that they are actually able to consciously and voluntarily elevate and depress their testis within the scrotal sac. Right? Just look at all the fun party tricks that you can show off the next time you get to socialize with other individuals. All right. However, and that's what I wanted to talk about next. I need more space. All right. So questions on these? Shockingly, with the reproductive lecture, when we start talking penises and vaginas and scrotums and things like that, the class goes terribly, terribly silent. And that's when we're all there and have the awkward eye contact that we have to make. Here, where we can all hide in our beds while we're taking this lecture, I expect it to be relatively quiet. But let us go ahead and sneak one more picture in here. I'm going to cover up this part, and I'll go ahead and actually pop that onto the screen there so we can talk about that. There is a third structure that helps us to regulate the scrotal temperature, and that is what is known as the Pampiniform Plexus. Basically what we have, and let's start with red and make it big. That's not very big, but that's all right. Actually, let's cheat. Undo. I want that. There you go. Excellent. So this is our testicular artery. And it's the gonadal artery. And if you remember way back in what seems like ancient times now, the beginning of this class when we were actually inside the classroom, we talked about those gonadal arteries and gonadal veins in males and in females. And one of the things we talked about is that males' gonadal blood vessels are much larger and more elaborate. One, because they have to travel farther outside the body versus inside the body. Two, because the testes are much more metabolically active than the ovaries are. If you think of the number of gametes that a male produces in a single day versus how many gametes a female produces in an entire month, it is dramatically different. So not surprisingly, there is a large blood supply going to the gonads. And so, of course, over here is our heart. And over here is our testis. But if you also remember when we talked about our cardiovascular system, one of the things we talked about our blood is 2 to 3 degrees warmer than our core body temperature. Our blood is hot. So we have this really, really hot blood that is going towards our testes. Think about it, having them outside the body, having these muscles that can relax and distend the testes further from the core of the body temperature doesn't do us any good if we have this massive flow of really hot blood coming to the testis that would dramatically increase our core body temperature, right? Again, just think about what happens when you're embarrassed as you're sitting there on Zoom and your significant other or the random guy that you met on some chat group is dancing his cremaster muscles for you. Oh, good gravy. My daughter's AP class always loves to send these voice recordings to her and the parents every morning on Monday, so we always get these fun phone calls. And you get embarrassed and all that blood flows to your face. Think of how hot your face gets when all of that happens when you're embarrassed. Well, we have the same issue here. We have this hot blood going to the testes, and that is a problem. Luckily, and I'm going to draw it as a single line, but what happens is we have this elaborate venous structure, and I'll cheat and draw a little of it this way. This elaborate network of veins that form this elaborate network over and that drape over our testicular artery. And if you think about it, one of those fancy words we've used for an elaborate network is the term plexus. And so what we have is this Pampiniform Plexus that is our venous blood that is traveling back to the heart. Of course, as we know, arterial blood flows away from the heart. And let's undo that. I don't like that. Black, small. There you go. Our arterial blood moves away from the heart systemically. Venous blood flows back to the heart systemically. So what happens is that we have, if you notice that term we used in the urinary system, a counter current mechanism. And in this case, that counter current mechanism forms what we call a heat sink. Right, this is an engineering concept that is used in many instances. If you have something that is warm temperature moving in one direction, what you can do is have cold water flowing back across it in the opposite direction, and it will steal its heat. And that's exactly what happens in this case. We have our venous blood traveling back towards the heart, and as it travels back towards the heart, it steals the heat from the arterial blood. So our venous blood steals the heat from the arterial blood. So what happens is when the blood reaches the testis, it is now cool blood, and it's able to fire the oxygen, the nutrients that the testes need. That cool blood then travels back towards the heart, stealing the heat on the way back, and so our venous blood is warmed back up. On its way back, and that cools the blood going to the testes in that way. So once again, we have another countercurrent mechanism, in this case forming a heat sink, that is going to help to maintain appropriate interscrotal temperature. And this elaborate network of the, again, all this really is in reality is the testicular veins. But these testicular veins for a very important, very elaborate structure that I guarantee you're going to have to spell at one point on the exam, that Pampiniform Plexus. Here we've identified these things in words then. So now again, I've got this great picture that we have done, but let's go ahead and clear all of that drawing. Here we have those three structures that are going to help to keep the testes at the appropriate temperature for producing viable sperm. And here we have the pretty picture that shows that as well. Notice we've removed that pesky penis to get it out of the way so that we can see these structures more elaborately. Here we see again, each testis is within its own pocket inside of the scrotal sac. They don't get to switch sides. On Monday you're on the left, and on Wednesday you're on the right. They are contained, as we can see, within that spermatocord that we see wrap around in it. Inside that, we see the cremaster muscles. These are those bands of skeletal muscle that can elevate or descend the testis within the scrotal sac. Notice within the wall of the scrotum itself, we have that layer of smooth muscle, that is the dartus muscle, that can change the shape of the scrotum. And then lastly here, we see how that testicular vein forms this elaborate network, right? That's what a plexus is, an elaborate network that drapes over that testicular vein, that testicular artery coming down to the testis, acting as that countercurrent mechanism, acting as that heat sink to steal the heat from the arterial blood, to cool that blood as it travels to the testis. All right, questions on any of that? Stunned silence. Excellent, just what I like during the reproductive system. All righty. Now, as we talked about in here, here we get a great view of that. Notice here we see the double layer of serous membrane that we talked about, the tunica vaginalis, with the visceral layer on the outer surface of the testis, and the parietal layer forming a pocket around it. That pocket, of course, is going to be lined with a thin layer of transudate inside. But when we talk about the anatomy of the testis, there is actually a second layer as well. So if we think of the structure of the testis, here is the structure of the testis. As we talked about, I'm going to do this in the highlighter just so it's going to be easier for me to do. We have that visceral, and I'm doing a horrible job of drawing it, which is exactly what I did in the highlighter. Excellent. We have that tunica vaginalis, or more specifically, the visceral layer of the tunica vaginalis that surrounds the outer layer of it. But the bulk of the testis, and now I'll switch back to black with the line, is this fibrous, and ironically I'm using black, this fibrous white connective tissue, which is why it gets the name tunica albuginea, right? Albino means to lack pigment. This is a whitish color fibrous connective tissue that not only forms the bulk of the testis, but there are also extensions of this tunica albuginea that help to divide the testis into individual lobules. And that's what we're seeing here. These particular invaginations of the tunica albuginea form structures that we call the septa. Septa, of course, being the singular, septum being plural. And since I use the plural, let's go ahead and draw two lines. The septa there that divide the testis into individual lobules. Inside this lobule, as we can see from the illustration, is going to contain anywhere between, and I'll sneak it in down here because it's going to take a little bit more space to write it, anywhere between 1 and let's say 4 small tubular structures that are specialized for making the sperm, and those are our seminiferous tubules. So inside these are going to be 1 to 4 of these twisty-turvy tube-like structures that are the seminiferous tubules. Let's go ahead and clear that. So again, the septas are the extensions of the tunica albuginea that forms the testis into lobules, anywhere between 250 and 300 lobules. Inside of them are anywhere between 1 to 4 seminiferous tubules. So again, if you think about it, 300, we'll keep the math simple, 300 lobules, 4 seminiferous tubules per lobule, right? That's as many as 1,200 individual lobules. And these seminiferous tubules have some smooth muscle cells wrapped around them that we'll talk about and identify in a minute. But each one of these seminiferous tubules is about 3 feet long. So if you think about it, right, 300 lobules, up to 4 seminiferous tubules, each one is 3 feet long. So if you were to take them out and lay them end to end, it would hurt a lot. There's a question. If the scrotal cavity is the same as the cavity of the tunica vaginalis? No. The scrotal cavity is the space inside of the scrotal sac that contains the testes. The testis itself is surrounded by the cavity of the tunica vaginalis because, again, it is a double layered membrane. So if you go back here, the scrotal cavity would surround the entire testis and the epididymis and the scrotal sac and some of these structures, whereas the cavity of the tunica vaginalis, as you can see, is just this space between the visceral and parietal tunica vaginalis that we see just around the testis. All right? Good question. So any others? All right. So again, miles worth of seminiferous tubules in the male's testes, and the function of these seminiferous tubules is basically production of our spermatozoa. A sexually viable male produces, a billion sperm during the course of the day, or some crazy number like that, right? Now, the seminiferous tubule itself, remember, we talked about this a little bit during the endocrine system, does not produce the hormones. But remember, within the lobule and within the septum, in the spaces in between are those interstitial cells. And remember, those interstitial cells that are outside the seminiferous tubule, but obviously contained within the testis, within the tunica albuginea of the testis, we have our hormone-producing interstitial cells. Here we see the light microscopy view of this, and here we see a pretty illustration that shows this same anatomy of a cross-section through a single seminiferous tubule. And there's some important things that I want to show you on this illustration that we'll continue to talk about. First, as we just finished mentioning, those interstitial cells, or the interstitial cells of Leydig, remember they were called, are the cells that produce the androgens. They are located outside of the seminiferous tubule. They're not contained within the seminiferous tubule. As I also just mentioned very briefly, but I will emphasize it again, notice that the seminiferous tubule is surrounded by a single layer of smooth muscle cells known as the myoid cells. And I notice as I look at this illustration, they don't have a label on that. So I'll cheat and go back half a slide, right? These myoid cells are smooth muscle cells that surround the seminiferous tubules. So that's what this is. This is a single layer of smooth muscle cells on the outer surface. And it looks a lot like a circular layer of smooth muscle, which indeed it is. And as we know, circular layers of smooth muscle change the shape of the lumen of a tubule. And if you change the shape of the lumen, you change the shape of the volume of the lumen, which changes the pressure, which can cause the fluid and the sperm to move. Which doesn't make a lot of sense because, as we know, sperm are the only cells that are capable of propelling themselves. But as it turns out, spinning that tail to propel yourself through space requires a tremendous amount of metabolic energy, a tremendous amount of fuel and resources. And so to allow the sperm to slowly swim their way through the male reproductive system before being released in part of the ejaculate would be a tremendous waste of metabolic resources. So as it turns out, the sperm themselves don't actually start their swimming motion till they are mixed with the fluids of the ejaculate and expulsed during ejaculation. What that means is to move them through the ductwork system of the male from one location to another requires things like fluid production and fluid pressure, and like here, smooth muscle contractions to change the pressure and cause the fluid that contains those sperm to be moved through the seminiferous tubules. So it's actually going to be contractions of this myoid layer, which is going to propel the sperm once they're finally made out of the scrotum out of the testis and through the ductwork system to be ready to release as part of the ejaculate. Notice, and we'll spend a lot of time talking about this, one of the things that we can also see is the variation in the shape and the size of these cells within the seminiferous tubules. One of the convenient for you things about this is that as the cell matures from a stem cell, and more specifically a cell-shaped stem cell, all the way to a sperm-shaped sperm cell, they mature and move from the outer wall of the seminiferous tubule towards the lumen, where they're finally released into that lumen. And so not only their size, but their relative position will be able to tell us and be able to identify what these cells are. So on an illustration like this, I could point at any of these blue cells you see, and you should be able to identify what stage of development that is for those cells. But notice one last thing. Notice that not all the cells inside this seminiferous tubule are blue, or black, I guess, for that matter. There are these purple cells as well. And let me make my highlighter to emphasize this point. Notice these purple cells have elongated and kind of triangular shape nuclei to them, which is a very common characteristic that is easy to see. And these cells are called nurse cells, Sertoli cells, or sustentacular cells. Again, there's something we briefly talked about when we were talking about the endocrine system, and we'll talk a lot more about now. And these nurse cells, as the name nurse might indicate, plays a huge role in both the maturation of the sperm and also the protection of the sperm as they're being developed as well. So we'll talk about those in depth as well. So here we see a seminiferous tubule, and again, outside that seminiferous tubule are those interstitial cells, the interstitial cells of a laid egg, that produce our androgens. Here we see the same thing in actually a light microscopy view. We see cell-shaped cells becoming sperm-shaped cells. We see that thin layer of myoid cells on the outer surface here, some interstitial cells that are outside the seminiferous tubules. And notice we have this kind of elongated, almost triangular shaped nuclei that are the nurse cells that we can see in there as well. And these are things that you will need to be able to identify both on an illustration and also in a histology slide as well. And maybe we'll do another histology. There's a big, huge histology component of this. Maybe we'll do another histology review on Friday. We'll see how the week goes. But I think a lot of people thought that that was useful last time, so maybe that's something we will do again. And just to beat the dead horse, here we have an electron microscopy view of a seminiferous tubule, blood vessel on the outside, cells, and here we can see those flagella, the tails of our sperm here at the center of our lumen. Now, to be able to produce billions of sperm cells over the course of their life, obviously we need stem cells that are going to be able to do that. And in males, they have, and again, these are going to be unipotent stem cells. Remember, unipotent stem cell is a stem cell that can divide many times to produce new cells. But all the cells become one and one thing only, and those are eventually sperm or spermatozoa, and those are the spermatogonium, plural being spermatogonia. No, that's what I want there. And like I said, they can produce over 500 million sperm during the course of a day. As I mentioned, they're surrounded by these Sertoli cells, or what are also called the nurse cells, or sustentacular cells. Here, I like this illustration in purple because it kind of shows them, but I also really like this illustration from your textbook as well. Here in, I don't know, orange, I guess now, is our nurse cell. And what I want you to notice about this nurse cell is that, like I said, it has kind of a triangular or diamond-shaped, elongated nucleus to it. It is going to surround and support the sperm as they're developing. But notice also, they're going to form tight junctions and gap junctions with the neighboring nerve cells. The reason this is important is because what our nerve cells are going to do is they're going to help to form what we call the blood-testis barrier. Remember, the job of our immune system is to protect us from cells that aren't us. And as we'll talk about, gametes are cells that are very different from us. We wouldn't want to take one of our somatic cells and one of your significant other somatic cells and put that together to make a person, right? They'd have 3 heads and be able to see through walls or have all sorts of other weird things going on with them. So our gametes are very specialized cells, which is awesome for producing offspring, but it's exactly the kind of thing that your body wants to protect itself from, because it's something that it doesn't recognize as being us. So one of the important functions of these nerve cells is to protect the developing sperm from our immune system, forming that blood-testis barrier. So it is the nerve cell that provides the nutrients. It is the nerve cell that provides the chemical signals. It is the nerve cell that is that controls the maturation of the spermatozoa directly. As we know, right, what was the hormone again that stimulates sperm production? Anyone remember? I know all of us wanted to kind of blank out and forget the endocrine system like it was a bad hangover that can be forgotten. But does anybody remember the hormone that we said is responsible for sperm production?
Follicle stimulating hormone?
Follicle stimulating hormone is an important hormone, and it absolutely is the one that is responsible for producing, among other things, sperm production. Absolutely. Right? That is what produces the production of sperm. However, we did cheat a little bit. When we talked about follicle stimulating hormone, we said that it stimulates sperm production, and that is true. But what it actually does is it actually stimulates the nerve cells. And then the nerve cells then stimulate the sperm production. So we weren't incorrect in saying that follicle stimulating hormone stimulates sperm production. We just skipped a step in the process. What follicle stimulating hormone actually directly stimulates are these nerve cells. And then it is the nerve. I'm sorry?
Is it luteinizing hormone?
Well, remember, luteinizing hormone is the one that causes stimulates the interstitial cells to be able to produce androgens. So remember, it's going to be our luteinizing hormone that stimulates the interstitial cell here seen in green to produce our androgens. Those androgens are going to play a role in the maturation of sperm. But again, it's not wrong to say follicle stimulating hormone is the one that produces sperm. We just skipped a step when we described it the first time. Follicle stimulating hormone actually stimulates the nerve cells, and it's the nerve cells then that directly stimulate sperm production. So it is follicle stimulating hormone that causes sperm production. That was absolutely correct. But it's by the way it does it is by actually stimulating the nerve cells. I think we talked about this a little bit at the time because if you remember, follicle stimulating hormone actually really does 2 things. It stimulates the nerve cells to mature the sperm, but it also stimulates the nerve cells to produce inhibin. Remember, we talked about how inhibin is an important hormone that helps us to maintain appropriate sperm production. And we'll talk about this process in much more depth a little later. Today, maybe, probably next class, seeing how far we get on that. All right, perfect. So our Sertoli cells form that blood-testis barrier. Super important, super important. And When stimulated by follicle-stimulating hormone, they support the sperm development. They produce the chemical signals that cause the sperm to divide. They provide the nutrients necessary. And as we look at this illustration, we go from this big, huge cell-shaped cell to this sperm-shaped cell. And a sperm-shaped cell is really just a chromosomal missile. When it comes to the fertilization of an egg, or the production of an offspring, let's say it that way. When it comes to the production of an offspring, half the chromosomes come from mom, half the chromosomes come from dad. But when you make that first cell, and anybody know what the fancy name we call a fertilized egg?
Zygote?
A zygote, absolutely. If that zygote is going to become a person, that zygote as a cell has to have mitochondria, has to have rough endoplasmic reticulum and Golgi apparatus, and all of those fun things that a cell needs to be able to divide. And it gets none of those from dad. This sperm literally is a chromosomal missile. Dad's responsibility to the production of that zygote is chromosomes and chromosomes only. Mom has to provide chromosomes and all the other cell stuff necessary to be able to survive and divide. So notice there's a tremendous shedding of material during this process. And there it is. That shedding of that material then is going to be phagocyzed by these nurse cells, because after all, they're nutrients, they're amino acids, they're lipids, they're things along those lines, and they can then be provided as nutrients to the next set of sperm that is being developed as well. So part of the way it's able to provide so many nutrients for these growing sperm is by recycling those nutrients that are being shed from the sperm during that developmental process. The nurse cells also produce the fluids, because remember, it's these fluid pressures. Here's that myoid cell out here that's going to contract, producing that fluid pressure to help to propel the sperm, because remember, the sperm aren't swimming on their own. And the nurse cells are responsible for producing hormones like inhibin, which we already talked about, but also they're responsible for producing a special protein called androgen binding protein. Again, if we think about it up here, where's my text? There it is. We have those interstitial cells. And as we talk about, they produce androgens. But if you remember, androgens are steroid hormones. They're lipid-based. They're made up of cholesterol. And as we know, lipid-based things don't like water. So while we produce, and I'll cheat and draw it as a little square right here, this interstitial cell is producing these androgens. These androgens, being lipid soluble, aren't going to be affected by that nurse cell's plasma membrane, that barrier, so it's going to be able to enter into the lumen by diffusion, right? Some of it is going to diffuse into the blood supply so that it could be released throughout the body, but some of it is going to diffuse into the lumen here. Notice this androgen doesn't have any effect on the development of our sperm, but androgens do play a role in the maturation of the sperm. They're not needed to make sperm-shaped cells, but to make that sperm-shaped cell, it is necessary. So what happens is some of those androgens can float here into the lumen of our seminiferous tubule, because we want it transported out of the testis so that it can be with the sperm when the sperm are going to mature. So what our nurse cell is able to do is it is able to make these special proteins called androgen binding proteins that are capable of binding to androgens and making those androgens water soluble so that they can be transported to other parts of our ductwork system so they can be present when the sperm need them to mature. So these nurse cells, like most nurses, are underappreciated and do a crap load of work. All right. Questions on that? But at least the pay is good. All right, excellent. All right. We have now reached that magical point in your life where, as a student and an instructor, we need to talk about the birds and the bees. We are going to talk about the process of how we make gametes. We'll do this for the male, we'll do this for the female. These are absolutely processes you are going to be responsible for, and not surprisingly, this is a process that hopefully most of you are aware of called meiosis. Now, meiosis is very closely related to, but also different from, the process of mitosis. Excellent. Which you all should remember from 430. On the offest of chances that you don't remember mitosis, then I have some good news. The first is that we are going to go over it very briefly and quickly. But more importantly, we are about to take our first break, so you have 10 minutes to run to your textbook, grab chapter 3 where it talks about cells, and reread mitosis to make sure that it makes sense to you. Because when we come back, we are going to, in the quickest of quickness, talk about mitosis, remind ourselves of mitosis, so that more importantly, we can relate it to meiosis and see how it is similar and how it is different. All right, we are going to go ahead and take our first break here. We will restart at, my watch says 9:08, so let's come back at 9:18. And I will start the recording at that part. So any questions before we take our first break? All right, run and get your textbook, look at mitosis, make sure you're familiar with it, and I'll meet you back here in 10 minutes. So, as I mentioned, our goal is going to be to produce gametes. As I also mentioned, we're going to need to do this in both boy-type people and girl-type people. We are starting with the boy-type people process first. And in boy-type people, the process of producing gametes is a process called spermatogenesis. Spermatogenesis is the entire process from stem cell to sperm-shaped cell. This entire process involves 3 important steps or 3 important stages. The first is our process of mitosis. Mitosis is how we produce new cells. Again, if they're going to produce, close to 1 billion new sperm every day, we need to be able to produce new cells to be able to do that. And mitosis is the process by which we do that. Then we need to undergo the specialized process of meiosis. Meiosis is how we get from cell-shaped cells to sperm-shaped. Well, let me rephrase this. Meiosis is the process where we halve the number of chromosomes. After all, if you and your significant other are going to produce an offspring, you want to give them half of your genetic material. Your significant other needs to provide half of their genetic information to make one person with some random combination of all of our genetic information. And that is the process of meiosis. After the process of meiosis, we then have to turn those cell-shaped cells into sperm-shaped cells. And that is a specialized process known as spermiogenesis. Your book does an excellent job of describing the entire process of spermiogenesis. In the interest of time, I'm not going to hold you responsible for that on the exam, but it is useful information, and so knowledge for knowledge's sake is always interesting and important. So if you are interested in that, I encourage you to read it. We will do an overview of why spermiogenesis is important and what happens as a result of it, but we will not be going through it step by step. We will be doing the first two stages of spermatogenesis step by step, though. So it is important, but you won't be step by step for spermiogenesis, but obviously you're responsible for that name and being able to spell that name and knowing what occurs during that specialized process as well. Now, as always, as we've talked about, and really this was from the very first lecture in 430, one of the things we've talked about is that when you're learning biology, it's like learning another language. We said that is especially true in anatomy and physiology. And the other place we said that is especially true is in genetics. In genetics and in anatomy and physiology, it is like learning a new language. And so vocabulary is important. And reproduction, of course, combines those two, genetics and anatomy and physiology. So we need to make sure we understand some key vocabulary terms. The first is to understand what we mean when we say a diploid chromosomal number. This is a fancy way of saying the number of chromosomes you have in a typical body cell. Now, of course, one of the important things to understand is every living creature has a diploid number of chromosomes if it divides sexually. So that means plants, that means, bullfrogs, that means everything in between. And everybody's going to have a different number. I think we already mentioned it here, but remind me again, how many chromosomes do we as human beings have as our diploid number of chromosomes?
46.
46, absolutely. 46 total chromosomes. Right? Di, of course, means two. And the reason for that is we have those 46 chromosomes, but really what we have are 23 pairs of chromosomes. If you remember, back in ancient times, and by ancient times I mean in 430, we learned all of the anatomy that goes along with chromosomes. We know that our chromosome, and this is horrible, but it's all right. Our chromosome is comprised of DNA and proteins that work together to form a loose thread-like material we call chromatid. We know that when you tightly pack that chromatid, it becomes a chromosome. All right, that is some of the vocabulary that we learned. So here we have a particular chromosome. Of course, when they're tightly packed to become chromosomes, we have already replicated the DNA, well, really, let's say the whole thing. We've already replicated the chromatid so that we have two copies. And so we have two identical, and I know my drawings won't be identical here, but you get the point. Two identical copies, and of course these identical copies are called the sister chromatids. Oh, sorry. DNA and protein loosely is, I gotta cheat. Chromatin. Chromatin is our loosely packed DNA and proteins. We pack them up. I did it there too. Damn it. Don't laugh at me. I heard that. I haven't had enough coffee this morning. There we go. Chromatin. It becomes a chromosome. We have our two sister chromatids. You know, I'm not really mad. You are allowed to laugh at me. I don't care. Sister chromatids. And of course, as I'm sure you remember, those sister chromatids are held together by a special protein. And that special protein is a centromere. And when they're held together by a centromere, these two sister chromatids, and that's a C, collectively are known as one chromosome. Lastly, remember when we're thinking of terms of anatomy, to move these chromosomes around in space, each sister chromatid has a special protein. No, don't want it to be red. I want it to be green. A special protein on the outer surface of it that allows it to connect to the microtubules and be moved around. And remember, these are the kinetochores. All of this vocabulary should be familiar to you. If it is not, I do not kid when I say go back to chapter 3 and look at mitosis, because you definitely need to understand the process of mitosis to make sense of all of this. All right. So what you are looking at here in this incredibly elegant drawing that I've done is a single chromosome comprised of genetic material, 2 copies of that genetic material held together by a centromere with those kinetochores to move that around. And what happens is that you have 2 of them, pairs. And with those pairs, one from mom and one from dad. These pairs, as we'll talk about, are what we call homologous pairs. What that means is that they contain the same type of information. And let's just go ahead and draw a second one here. That is the homologous pair. And we'll do the centromere in the center. And we'll do the kinetochores on the outside. With these homologous pairs, these contain the same type of information. With the same type of information, what I mean by that is that this part of this chromosome may, for instance, be the part that determines handedness, meaning whether you're left-handed or you're right-handed. And that same component is going to be found over here, oops, highlighter, on that same location of that same chromosome there. And of course, will be on both pairs. Now notice I said same type of information. For instance, over here from mom, you may have gotten the right allele, the right version of that chromosome, the right version of that gene. But over here from dad, you may have gotten left. So it is the same type of information, but not necessarily identical information. And that is the key. We have 23 pairs of chromosomes. Each one, for the most part, has the identical information on both of them, types of information, but it may be different versions, right? Maybe down here is handedness, is blood type. You got A from mom, you got B from dad, and so your blood type is AB as a result of that. Right? So again, it could be different types of information, but the same type of information. And now I got to clear all my drawings there. So we have those homologous chromosomes that collectively form a diploid number. And here's the other key to this. So I guess I have to release all of this stuff individually because I want to keep this up here for now. The reason all this is important is because when we talk about our diploid number is typically indicated by the letters 2N. The 2 obviously means that it is paired, meaning that you have 2 versions of it. And the N is the unique number of chromosomes you have. So you have 23 unique chromosomes, and you have 2 copies of each of them. So your diploid number would be 2 times 23. 2N number is 46. All right. You have 46 chromosomes. You want your offspring to have 46 chromosomes. But you don't, so you need to cut your chromosomes in half. And that haploid number is obviously N. Now, you don't want to take your 46 chromosomes and put them together just randomly. Take random 23. Think of it this way. If you had a deck of cards and your wife had a deck of cards and you wanted to have a functional deck of cards, you wouldn't just randomly take 26 cards out of your deck, randomly 26 out of your wife's deck, put them together and have a functional deck. The easiest way to do it is take all the half of them from me, say like all the spades and all of the clubs, all the black cards from me and all the red cards from my wife, and we put them together, right? That way we have matching sets. So when we produce a gamete, we want that gamete to have 1 copy of each unique chromosome. And that way only will we be able to produce viable offspring. So our somatic cells are all going to be diploid in number, and we want our gametes, or what are also called our sex cells, to all be haploid in number. All right, with me so far? Excellent. And so again, the only haploid cells are those sex cells. We already talked about and identified what a homologous chromosome is. Again, those are the 2 copies of chromosomes, one you got from mom, one you got from dad, that have the same types of information. And so we call those homologs. We also talked about, and I drew the anatomy of a chromosome, which is 2 sister chromatids held together by a centromere. And we want a way to be able to distinguish, because again, here's where our vocabulary gets a little tricky. 1 copy of chromatin is a single chromosome. 2 copies of chromatin that are held together by a centromere is also 1 chromosome. So this is 1 chromosome. And this is 1 chromosome. So we need a way to distinguish this one where it's replicated versus this one where it's not replicated. So this one is not replicated, and this one here is replicated. So this one that is replicated, we refer to as an R, and this one that is not replicated, we would just refer to as N. So basically, that denotation of R means that it is a replicated copy of the chromosome. And so that's going to help us as a placeholder when we look at this process. All right. So that is our vocabulary. There isn't a lot of new vocabulary there. So hopefully that was all pretty simple and straightforward to you, except with the homologous stuff. So now what we need to do is talk about the difference between mitosis and meiosis. To do this quickly and easily, I'm going to cheat and move our meiosis from this illustration. And notice what we see here is our goal of mitosis. Our goal of mitosis is twofold. We want to divide one cell into two cells. That is the simple definition of what our first goal of mitosis. And again, it really should be plural goals of mitosis. Goal one, divide one cell into two cells. But the second part is we want the cells to be identical to each other and to the original. So that is our goal of mitosis. Our goal of mitosis is to divide one cell into two cells, and it takes one division to do this. That's a 1 division to do this. And at the end of this process, we want to have two cells that are identical to each other and identical to the original. Notice our illustration does a nice job of showing this. Here, our illustration clearly is not a human cell, but we wouldn't want it to be a human cell because we would need 46 chromosomes here in this tiny little space. Instead, notice they've given us 6 chromosomes, but they have shown us that these 6 chromosomes are 3 homologous pairs. So these two are long and blackish in color. These two are short and reddish in color. These two are long and bluish in color. So we have our single chromosomes. Notice these chromosomes are first replicated. And again, one of the important things to remember is that replication occurs.
You can do that side. Hey, look at me.
Not that, not the other side.
I get it. Start here. Work this.
So
6 plus 7.
remember, replication occurs in interphase. Replication is going to occur in interphase before mitosis begins. And so that's what we have here. Here we have our replicated. So again, as we mentioned, as a placeholder, we have cells that are now 2NR. They started as 2N. Now they're replicated, they're 2NR. And our goal is to have two cells that are now diploid, 2N, at the end of this process. And as hopefully you recall from 430, mitosis occurs in 4 stages. And again, yes, sometimes 5, depending on how you think of it. And of course, those 4 stages are prophase, metaphase, interphase, anaphase, and telophase. Prophase sometimes is divided into early and late, or sometimes, remember, we just have prophase and they've taken late prophase and separated into its own phase, prometaphase. But for simplicity, we'll just go with the 4 stages: prophase, metaphase, anaphase, and telophase as we talk about those. Briefly, to remember what happens during these phases in prophase, basically there are 3 main events that begin, well, let's say in early prophase, 3 main events that begin. Those 3 main events that begin are, first, they are the packing of the chromatin into chromosomes. So we start the packing of that up. The second thing that happens is our nuclear envelope starts to break down. And the third thing that happens is that, remember, the trigger to start mitosis from interphase is the development of that second centrosome. Our two centrosomes start to migrate to the opposite sides of the cell. These are the three events that occur. They start in early prophase, and by late prophase, all three processes are completed. And more importantly, on top of that, well, not more importantly, but also our spindle fibers have formed. And remember, the spindle fibers are microtubules. And those spindle fibers form and they connect to the chromosomes. Once that occurs, we end prophase and we begin metaphase. Metaphase is where the chromosomes are moved through the cell to the equator, or the metaphase plate, which is the equator, between the two centrioles of the centrosomes that are there. And remember, the key with mitosis is that all chromosomes line up independently. So they all line up on that metaphase plate. That triggers the beginning of anaphase. And anaphase, the centromere inactivates. And when that centromere inactivates, the now sister chromosomes, oh, let me say that again. All right, so let's say it this way. Our centromere inactivates, and when it inactivates, our sister chromatids become daughter chromosomes. And then those daughter chromosomes are pulled to the opposite sides of the cell. And then once they're pulled to the opposite sides of the cell in telophase, basically everything that happened in prophase happens in reverse, right? Our chromosomes are going to unwind into loose chromatin. Are now two nuclear envelopes, whereas we had one nuclear envelope before. Now two nuclear envelopes will form around the two new nuclei that form. The two new nuclei, I guess that's good. And of course, remember also in telophase, or during telophase, but actually starting back in anaphase, we also have cytokinesis, where we split the one cell into two cells. Right, so the one cell is divided into two. Again, very brief explanation, but it is important to understand these steps because we need to understand how mitosis is going to be different from meiosis when we talk about it. For starters, meiosis is going to use 2 divisions instead of the 1 that we're going to see in mitosis. But even more importantly than that, there are going to be some differences, especially in prophase and metaphase, that are going to be different in meiosis than they are in mitosis. Again, I appreciate this is a very, very quick introduction to this, but it is something you should have recalled from 430. And if not, I encourage you to go back to your textbook's got some great resources that talk about mitosis. And then if you guys are really, really struggling on it, like I said, we'll do a Friday review on histology again. And on Friday, we can also do a mitosis review as necessary as well. All right. If we have a basic understanding of what's happening in mitosis, then what we can do is talk about what is going to happen during meiosis. So let's go ahead and clear all of that and see how meiosis is different. In meiosis, and let's start over here, our goals, plural, are different. Whoops, I need that to be black. There we go. Our goals are different. We still have 2 goals, but our goals are different this time. This time, goal 1 is to divide the cells twice. And in this way, what happens is we go from 1 cell to 4 cells. So that's our first big difference between mitosis and meiosis. But we have a second goal as well. The second goal is really twofold. First, we want to make cells that have half the number of chromosomes. All right, we want them to be haploid. And let's go ahead and make it as a third goal, just instead of putting them together. We want the new cells to be unique, not just, oops, there we go. Unique, not just from the original, but also from each other. So we want 4 completely unique cells that are different from the original and different from each other and have half the number of chromosomes. So that is our goal. To do this requires not 1, but 2 divisions. So when we look at those 2 divisions, the first division, not surprisingly, is called meiosis I. And again, typically Roman numerals are used for this, but if you want to use Arabic numbers, that is fine as well. Meiosis I is the one where that is most different from mitosis. Right? And the reason for this is during meiosis I, instead of the chromosomes working independently, remember in mitosis, every chromosome was for itself, did its own thing. During meiosis I, our goal is to connect and control our homologous chromosomes. And we'll see how that occurs in a moment. We do this so that at the end of meiosis I, we have haploid cells. And these haploid cells, as we're going to see, and you see that in the illustration to the right here, have replicated chromosomes. So if you think about it, here we start with cells that are 2N, and then of course, as they get ready to divide, they become 2NR, where that information becomes replicated. At the end of meiosis I, we want haploid cells, but haploid cells that are still replicated. So that when we have meiosis II, and I guess it should be capitalized for consistency, when we undergo meiosis II, this is essentially identical to mitosis. This process is essentially identical to mitosis, where each I hate having to type and talk at the same time. I can barely write on the board and talk at the same time. Having to type where I have no typing skills is just the worst. Essentially identical to mitosis, where each chromosome works independently. And at the end of the process, the sister chromatodes are split apart into new cells. So whereas we go, and let's be more specific here, up here we have one cell that is 2N and 2NR. To here we now have two cells that are haploid and replicated. And at the end of meiosis II, we now have 4 cells. And those 4 cells are all N, haploid and unreplicated. I'm going to bring up the whiteboard and I want to show you what's going to happen. I don't know how easy this is going to be to do. Actually, hold on a second. Let me do this first. I'm going to leave this here for a second because I'm afraid that if I stop sharing that these are going to go away. But I guess we'll risk it because we need to move forward anyway. All right, let me take a look at something. Okay, this works. You know what? Let's do that. Yep, I was afraid of it. It went away. Okay, that's fine. Rather than me trying to draw it, I think the easiest thing to do is, because again, me being able to draw this is going to be hideous. So I think the best thing to do is actually to go through the pretty pictures from your textbook. And I'll really make a point of emphasizing and pointing out the key steps and components in this process. So here is meiosis. And I guess the last thing that we need to say, as I mentioned, is we have 2 divisions. And those 2 divisions are meiosis I and meiosis II. And each of these processes have 4 steps. And so not surprisingly, they are prophase I, metaphase I, anaphase I, and telophase I. And meiosis II also has 4 stages. And those 4 stages, of course, are going to be prophase II, metaphase II, anaphase II, and telophase II. So what we are seeing here at the top of the screen is prophase I. This is the beginning of meiosis I. And remember, our goal in meiosis I is to organize the homologous chromosomes. And that's what we see happening. Notice at the beginning of this, this looks just like mitosis. Our loose chromatin is condensing down into our chromosomes. Those chromosomes are sister chromatids held together by a centromere. Our nuclear envelope is breaking down, and the centrioles of our centrosomes are moving to opposite poles. This would be early prophase. By late prophase, our centrioles have reached the poles of the cell, our chromosomes are formed, and our nuclear envelope has broken down. Everything is the same as before. However, what's special about prophase 1 is that as the chromosomes form, and I think I'll sneak it in here. What happens is that the the homologous chromosomes find each other. And when they find each other, they have a special protein. And that special protein is called synapsis, and that synapsis binds the homologous chromosomes together. So notice what we have here on our illustration right here. In yellow, we have mom's 1 chromosome. Here in yellow. Here in red, we have dad's 1 chromosome. Again, remember those chromosomes contain the same type of information, right? Up here is handedness, down here is blood type, down here is predisposition for heart disease or something along those lines. So the homologous chromosomes have found each other, synapsis binds them together, and they form a structure called the tetrad. If you think about it, this tetrad is 4 pieces of chromatin, all containing the same type of information. So again, with simple terms, and I'll use the highlighter for this, and we'll use purple, right? Right there could be handedness. So there's mom's right-handed and dad's right-handed, and dad's 2 copies. Again, there's dad's 2 copies of handedness there, mom's 2 copies of handedness there, and these things come together and form a tetrad. Our goal is to organize the homologous chromosomes. So in meiosis 1, first thing we do in prophase is we form tetrads. And those tetrads are the organized homologous chromosomes. Once we form those tetrads, then meiosis is all about those tetrads. Prophase is where we form the tetrads. And let me cheat and grab this and move it out of the way. Metaphase, we now line up the tetrads. The same way that in metaphase of mitosis, individual chromosomes line up along the center point. Now tetrads line up along the center point. So we form tetrads in prophase 1, we line up tetrads in metaphase 1, and in anaphase, we separate tetrads. What happens is that in this way, mom's copy goes in one direction and dad's copy goes on the other. And notice how they line up is a flip of a coin. So mom sometimes can be on the left side and mom sometimes can be on the right, right? It's a flip of a coin. This flip of a coin is vitally important because when you flip a coin, how many different options do you have?
2.
2, heads or tails. When you flip a coin twice to, how do we do the little V thing? Oh, there it is. 2 to the second power. How many different options do I have then?
4.
Right. Heads, 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 tails, tails, heads, tails, tails. 4 different times. When our chromosomes are lining up, how many tetrads are going to form?
23.
23. So each one of those tetrads is a coin that can be flipped. So if I have 23 coins and I'm going to flip them, how many different combinations can I come up with? What is 2 to the 23rd power? I'll wait while someone puts that into their smartphone, or you're sitting in front of a computer. Bring up your calculator there. What's 2 times 2, 23 times? Come on, we're not moving forward until someone gives me a number.
A lot.
It is a lot, but I actually want a number. There's a number. Excellent. And that is a beautiful number. Notice that is over 8 million different permutations that it can come up with. So if you think about this, just the way our chromosomes line up can give us over 8 million different combinations of how you produce gametes. And your significant other's doing the exact same thing. So you have your 8 million combinations that can go together with their 8 million combinations. That's a lot of variation, isn't it? Which is exactly why you look similar to your sibling, but you're not identical. Every time you make a gamete, it is going to be unique, right? You do not look like your siblings, and that whole Patty Duke thing is complete bullshit. And I know none of you get that reference, and I tell this joke every semester, and I don't care. That one is solely for me. If you don't know who Patty Duke is, I don't care. Look it up on YouTube after class. All right, excellent. And notice there's something else wonky going on this we haven't even talked about yet. We'll get to that in a second. But the point of this is that what Meiosis I allows us to do is to form our chromosomes for division like we normally do. But what we are able to do is take our homologous chromosomes and put them together, form those tetrads. And once we form those tetrads, we can be sure that when we make new cells, each one gets 1 copy of chromosome 1. Each cell gets 1 copy of chromosome 2 and chromosome 3 and chromosome 4, all the way through the 23 pairs. So that each cell is going to have the correct number of chromosomes and the correct genetic information. Because you don't want to take both copies of 4 and stick them on one side and both copies of 3 and stick them on the other. You could have 23 chromosomes that way, but you wouldn't have all the genetic material you need to make sense. And so then after telophase and, of course, cytokinesis, after those processes, we now have 2 haploid cells. But notice the chromosomes, the DNA, is still replicated. So we now have cells that are NR. Because we formed tetrads, we lined up tetrads, we separated tetrads, and then we isolated those tetrads into nuclei and divided them into 2 cells. Now, one last thing, and I'm going to cheat, and I'm going to sneak back to the previous picture. Notice when we look at this previous picture, when we look at the chromosomes, notice these 2 cells are identical to each other. This one got all of dad's stuff, this one's got all of mom's stuff, and this has these pieces of information, and this has these pieces. Notice these are different from the original. This is different from this one, but these 2 aren't unique, and these 2 aren't unique. Notice that doesn't totally accomplish our goal. So there is one other process that is taking place here. And this other process that is taking place also occurs during prophase 1. During prophase 1, we have a process called crossing over. Notice what happens with crossing over is that some of the genetic information from mom's chromosome will change places with the genetic information from dad's chromosomes, making each one of these copies. Remember before, as we look at it here, and where's my highlighter to emphasize this? Notice here, this one's all red, this one's all red. These two sides are identical. But notice now this one here and this one here are no longer identical thanks to crossover. Crossover increases the genetic variation of what we produce well beyond 8 million different possibilities. It truly makes every single gamete you produce absolutely unique. This crossover can happen anywhere between 1 to 20 times within a single chromosome, and that varies across all of the potential chromosomes. And it's what gives everybody their uniqueness. That's how you get grandma's nose and grandpa's ears, mom's blood type, but dad's predisposition to a heart disease. This basically allows for the random assorting of the genes, this crossover that occurs. And it is this crossover that truly makes every single cell unique. And we'll see that at the end of the picture of this process here. All right, so again, at the end of meiosis I is about tetrads. Prophase 1, we form the tetrads. Metaphase 1, we line up the tetrads. Anaphase 1, we separate the tetrads, so that at the end of telophase, we have haploid cells with replicated DNA. Well, taking a cell with replicated DNA and dividing up that DNA is exactly what we do in mitosis. And that's what meiosis II is. Meiosis II is essentially mitosis. So let's get my eraser and get rid of that and get rid of that. Here we see meiosis II. And in meiosis II is essentially identical to mitosis. One of the reasons you need to understand mitosis is you need to know what happens here. Really, the only difference is the number of chromosomes. In mitosis, you're dealing with cells that have 46 chromosomes. In meiosis, you are dealing with cells that have 23 chromosomes. But notice in prophase 2, we form the chromosomes. Metaphase 2, we line up. So our 23 chromosomes form here in prophase 2. Metaphase here, chromosomes line up. Anaphase, chromosomes are separated. And so then in telophase and cytokinesis, we form new cells. And notice here, they just showed it for one of these cells. It happened to be this one. So notice they just showed it for this cell right here, how it divided into two cells. But remember, this does it as well. And I believe the next picture does a good job of showing that. All right, so let's look at this process again. Questions? All right, let us look at this process again. Switch the screen. There we go. Clear all the drawings. So again, notice, and this is right out of your textbook. Your textbook does an excellent job of showing this. Notice this one had 4 chromosomes, 2 homologous pairs. We have the same thing here. 2 big, 2 small, green to represent mom, blue to represent dad, or the other way around, whichever you like. Meiosis I is about tetrads, right? Not only does our nuclear envelope break down, not only do our chromatids move to, our centrioles move to opposite sides, not only does our chromatin condense down into chromosomes, but homologous chromosomes find each other, and that special protein, synapsis, zips them together. So in prophase 1, we form our tetrads, and crossover occurs. Metaphase 1, tetrads line up. Notice this time, moms are all on the left, dads are all on the right, but it could have just as easily been the other way for each of these. Each of these was a coin flip. And notice, nice enough to put one crossover on this so we can see how that affects the uniqueness of these. So we had one single chromosome crossover that took place. So metaphase, our tetrads line up. Anaphase, our tetrads separate, so that in telophase, we form 2 cells, and cytokinesis, we form 2 cells with nuclear envelopes that have now half the number of chromosomes, but still replicated DNA. So that in meiosis II, and now we get to see both of them, prophase, chromosomes form, nuclear envelope breaks down, centrioles move to opposite poles, right? Chromatin condenses down into chromosomes, so we form our chromosomes. Metaphase, we line up the chromosomes. Anaphase, we separate the chromosomes. And telophase and cytokinesis divides them, now those 2 cells, into 4 cells. Each now of these are now N. So again, we went from these were cells that were NR to now cells that are N. So we have 4 N cells, 4 haploid cells. And notice when we look at the genetic information, this one has all moms, this one has mostly moms and some dad, this one has mostly dad but some mom, and this one all dad. Again, the key to this process is with crossover, we get 4 unique cells. All right, and those are our 4 cells. Oh, that should be good. And that is meiosis. Now here's the good news. This process of mitosis and meiosis, both of these processes occur in both men and women. So understanding mitosis, understanding meiosis is vital in both men and women, because the processes of mitosis and meiosis in both men and women are the same. However, gamete formation in men and women are not the same. So while meiosis and mitosis both occur in males and females, and they occur the exact same way, these exact same processes, the process of spermatogenesis and the process of oogenesis in females are going to be different. And so if we understand mitosis and meiosis, then we can finally go back to that process of spermatogenesis to understand specifically how this happens in males. And then probably on Wednesday, we will talk about how this happens in females. So questions on this? If this doesn't make sense now, it's only going to get worse. So any questions on this mitosis, meiosis I or meiosis II that I can help you to understand so we can move forward? All right, excellent. Stunned silence, what I always love. So let's now talk about these processes of mitosis, of meiosis, and specifically talk about them in terms of sperm formation. the process of spermatogenesis. As we said, spermatogenesis involves 3 stages: mitosis, meiosis, and spermiogenesis. This is different from females, right? Mitosis and meiosis both occur in males and females, but spermiogenesis is a process that is only in males. So that is something that's important to remember as we're going through this. But let's talk about these processes. In sperm formation, as we talked about, it starts with a stem cell, a unipotent and that unipotent stem cell is the spermatogonium. These stem cells are present at birth, but only become active at puberty. And as was already mentioned, it is that follicle-stimulating hormone that is going to cause the spermatogonium to divide by, as we talked about, stimulating the nurse cells to do that. Those spermatogonium are then going to undergo mitosis and become primary spermatocyte. Those primary spermatocyte are then going to start meiosis and become secondary spermatocyte. Secondary spermatocyte undergo Meiosis II and become spermatids. And let's look at the pretty picture to see how this works. Here's some vocabulary, but again, normally I'd be drawing this on the board, but I think in this case, it's easiest to go through it on the pictures from your book. So let's go ahead and do that. Here are the steps in the process. Again, this begins with mitosis. We have our stem cells that, when stimulated by follicle-stimulating hormone and from the nurse cells, because really it's the nurse cells that stimulate this process, they divide mitotically. And of course, as we know, when you divide mitotically, you produce 2 new cells that are identical to each other and to the original. All the spermatogonium, gonia plural, are located at the outer wall of the seminiferous tubule. So here we are at the outer wall of the seminiferous tubule. That spermatogonium divides as a diploid cell into 2 diploid cells that are identical. So again, it produces 2 identical cells. And these 2 identical cells are, as you can see, the type A and type B. Now, if they are identical, how can there be 2 types? Well, the easy example is really based on location, right? That's a concept that we are familiar with, that things can be slightly different based on their location. Typically, the type A cells are going to be closer to the outer wall of the seminiferous tubule, and I'm just going to abbreviate that as T now. And these cells are the ones that become new stem cells. If you're going to produce 1 billion cells during the course of a day, then what we need to be able to do is make more stem cells. When one cell divides to produce new cells, one of them has to replace the original. And so that's what happens. This basically replaces the original stem cell and keeps the process going. However, the type B cell that is produced is typically further from the wall. And this one matures to become the primary spermatocyte. And this primary spermatocyte is the cell that undergoes meiosis I. So here we see the two identical cells that are being produced. The type A stays here at the wall and it becomes a new spermatogonium, whereas the other one matures. And part of that maturation process is to get ready to divide. And this cell, remember, is going to have to divide twice. If we're going to divide and produce 4 cells, one of the things we need is lots of cytoplasm. So the cell gets bigger. And of course, the other thing that has to happen is we need to replicate the DNA. That makes this primary spermatocyte, really makes both these cells, the spermatogonia and the primary spermatocyte, easy to identify. The spermatogonia is at the outer wall of the seminiferous tubule, and the primary spermatocyte is the largest cell in the seminiferous tubule. It's going to be slightly away from the wall, but it is the largest cell in the seminiferous tubule, and so it makes it pretty easy to identify. So again, production of spermatozoa. So here we got all, whoops, out of control. Stop it. There we go. Excellent. So we have this. We've described all of that. Again, nothing new here, really. This begins, again, at the onset of puberty. With an increase in follicle-stimulating hormone. And when does sperm production end? When do males stop producing sperm?
About 10
They
minutes.
die?
after they die.
Never.
There you go. I like that. About 10 minutes after death. There you go. Exactly. All right. This is how you have plenty of people like Tony Curtis having kids way past the age when he should be having kids, because they produce that sperm basically till 10 minutes after death. It is a continuous process. There may, keyword there being may, there may be a slight decrease in sperm production and viability as we age, but it doesn't have to. So those old men can be just as fertile as the young boys are. Right? Excellent. Again, that spermatogonium undergoes mitosis. Follicle-stimulating hormone is the hormone that stimulates that, and we get those 2 cells. The type A that are going to stay spermatogonia, replacing the original cell, and the type B that grow and get bigger and become our primary spermatocytes. And it is those primary spermatocytes that undergo meiosis I. Notice in meiosis I, we get 1 division where 1 cell becomes 2 cells. But remember, meiosis I is all about the tetrads. So while we have 2 cells, the 2 cells are haploid in number, but still replicated. And if you notice now, if you look at the pretty picture over here on the left, this is why I like using that R. So notice our primary spermatocyte is really 2NR. Its chromosomes are replicated. So that when we form secondary spermatocytes, these are NR, and those haploid cells with replicated DNA then become just N. Notice otherwise, you don't quite understand how you go from N to N, right? Obviously from 2N to N, that makes sense. You cut it in half. But how do you cut it in half again and stay N? And the reason for that is it's replicated. So I like that R as a note, as a placeholder to remind us that this chromosomes are replicated at these two stages and not replicated here. So our primary spermatocyte undergoes meiosis I, producing 2 haploid cells, and these haploid cells are identical in that they're both haploid, but again, they're unique already in the type of genetic material they have. Our secondary spermatocytes then undergo meiosis II, and we get now 4 haploid cells. And the key also to remember here are these 4 haploid cells are still cell-shaped cells. And these cell-shaped cells are called early spermatids. So now we have 4 unique cell-shaped cells with a haploid number of chromosomes. But we still need to go from cell-shaped cells to sperm-shaped cells. So notice we've completed mitosis. We have now completed both stages of meiosis. But what we still have to do is that third step. And that third step is spermiogenesis, where we go from cell-shaped cells to sperm-shaped cells. As I mentioned, your book does an excellent job of describing this process. You can see it's an elaborate 7-stage process, which I encourage you to read, but will not be on the exam. So maybe read it during the summer for some fun light reading. But basically, the goal is threefold. We want to go from a cell-shaped cell to a sperm-shaped cell. We need to become specialized so that this cell is mobile. Again, this spermatozoa are the only cells that are capable of propelling themselves through space. And that requires a tremendous specialization to this cell. So the cell has to form these specialized structures, and it needs to shed a majority of its cytoplasm, which again will be phagocytosed by the nurse cells and used in the production of new sperm. So again, we have to undergo this process of spermiogenesis to make sperm-shaped cells. And at the end of this, we have late spermatids. Now, these late spermatids are now physically mature, meaning that they are physically, they are, sperm-shaped cells. But they are not functionally mature. To become functionally mature, they need an additional 20 days of maturation. And again, this maturation is with our androgens. Remember, we don't need the androgens to make them, but we do need for the maturation of this. And this maturation actually occurs outside of the testis in a structure known as the epididymis. There you go. In a structure called the epididymis. Now, notice this process of going from a stem cell, one stem cell, to 4 sperm-shaped cells takes about 64 to 72 days. We then need an additional 20 days of maturation. So if you think about it, from stem cell to viable sperm takes around 90 days, right? And you thought the lines at Disneyland were bad. Imagine waiting 90 days for a 3-minute ride, right? So we have this process, and at the end of that process, we have viable gametes. Now, as I mentioned, one of the things the sperm has to do is become specialized, and it has 3 specialized regions. No, I need that. Thank you. The first is the head, or what is called the genetic region. The genetic region is obviously where it contains the chromosomes. So it has its nucleus here. And remember that nucleus has its 23 chromosomes in it. But, you know, one of the phrases that I use in this class frequently, and again, you guys even listening to me online have gotten this, there are many turns of phrases that I use all the time, and a frequent one I'll use is a candy-coated shell. A lot of times when they have an outer thing, I refer to it as a candy-coated shell. Well, here, finally, at the end of the class, when we are talking about the reproductive system, the egg actually literally has a candy-coated shell. Part of the protections for our ova is a glycogen-rich hardened outer shell. It has a candy-coated shell. And so our sperm need to be able to break through those defenses. And the way it's able to do that is it has this special structure on the top of the head known as the acrosomal cap. And this acrosomal cap contains digestive enzymes that are capable of breaking down those glycogens to break down those defenses of the egg and get in there and fertilize it. There is nothing more American than the fertilization of an egg, right? Anytime you see this process in the movie or cartoons or anywhere they talk about anything else, it's always, you know, you see all the sperm racing to get there because the first one gets there, yay, wins the race. No. What happens is the first one that gets there, in fact, the first 200 that get there, have to work themselves literally to death, breaking down the defenses of that egg, so that that lazy slacker 201 sperm that comes lollygagging up after the 3-cocktail lunch and stopping to flirt with the secretary on the way is able to penetrate through those defenses after the others have killed themselves and fertilize that egg. America, right? So we have that digestive cap to be able to break down those defenses. We need that motility. What's interesting about the sperm, as I said, is it is the only cell that is capable of propelling itself in space. The reason it's able to do that is it has like a rotary motor inside of it. Basically, a rotary motor is a motor that rotates in a circle. And as it rotates in the circle, it whips the tail around in a circle fashion, in a whip-like motion. And that whipping of the tail is what propels it through space. Right? That rotary motion, every single notch of that rotary motion requires ATP. So here in the mid-piece, or what is known as the metabolic region, not only do we have the rotary motor, but what's represented here in blue, and actually if we cheat and we go back to the previous picture, we can see what's actually happening. Mitochondria tightly wrap themselves around that motor region. So right there, we have all the mitochondria necessary to make all the ATP to spin that wheel. And of course, the spinning of that wheel propels the tail-like or locomotive region, which is that long flagellum structure that again moves in a whip-like motion to propel the sperm through space. And so just that simply, we have that process. And again, here we have the pretty picture. Notice here with the illustration, we can get a sense of not just the process, but where we'll find these cells when we look at this histologically or on a model. Remember, as we talked about, The stem cell, the spermatogonium, is always against the outer wall of the seminiferous tubule, furthest from the lumen. It divides to produce two identical cells mitotically. And really, the only difference between these two cells is their location. The one that is closest to the basal surface of our seminiferous tubule becomes a stem cell. The one that's furthest matures and grows and becomes the largest cell in the seminiferous tubule, and that is our primary spermatocyte. The primary spermatocyte undergoes meiosis I, producing two haploid cells known as the secondary spermatocytes. The secondary spermatocytes both undergo meiosis II, dividing once each to produce a total of 4 cells, and these 4 cells are cell-shaped cells, are early spermatids. And then these early spermatids undergo spermiogenesis to become sperm-shaped cells by the time they reach the lumen. And that is our process of spermatogenesis. Questions on that? All right, excellent. This is another good stopping point. Let's go ahead and take our next break here. We will restart in 10 minutes, and it looks like that's going to be 10:47, and I will start the recording. All righty. All righty. So we've got most of the heavy lifting done and out of our way now that we have had a chance to talk about spermatogenesis. We have now gone through the process of making the sperm-shaped cell. And that is what occurs in the seminiferous tubule. As we said, then our goal is going to then be to move the sperm out of the testis so that it can be matured. If you notice, the seminiferous tubules empty into a structure called the rete testis. The rete testis is a mesh-like structure located within the testis. Notice, though, to exit the seminiferous tubule, because remember, one of the things we talked about, and let's go ahead and write this out. Let's do it up here so we have a little more room. The male duct work system is a closed duct system. So again, from the seminiferous tubule, there is only one place the sperm can go. And that one place the sperm can go, as you see right here, is this exiting structure that comes out of the seminiferous tubule called the straight tubule. The straight tubule feeds into the rete testis. The rete testis then exits the testis by a structure called the efferent ductule. And those efferent ductules then feed into a structure called the epididymis. As I said, whether you own them or whether you borrow them from someone else, hopefully you'll have the opportunity to explore the testis and scrotum of a male-type individual. When you do that and observe the raphe, when you see them move the Cremaster muscles to elevate and descend the testis, ask them to be able to palpate their testis. And if you do that gently, what you will feel is that the testis, as we said, is an oval-shaped structure that is about 1 inch by 2 inches in size. But as you can see here, there is an enlarged structure that sits on the testis. It starts superior. It goes posterior and lateral. So again, it goes from the superior along the posterior and lateral aspect of the testis on both testes. And that is this big, huge coiled structure known as the epididymis. So from the producing of the sperm out through here, is this structure. And again, remember, the sperm are not propelling themselves. Instead, we're using fluid pressures as the nurse cell produces those fluids, and then the myoid cells contract around the seminiferous tubule. It produces those fluid pressures, and then also our friend peristalsis will be used to move them as well. So again, it is going to be that hydrostatic pressure, right? Changes in fluid pressure of the myoid cells, out the straight tubule, into the rete testis. Again, at this point, remember, they are still not viable. They need to exit the testis through the afferent ductule into the epididymis, where that maturation is going to take place. As you can see, the anatomy of the epididymis is a tightly coiled tubular structure, about 23 feet long, that again starts superior and then goes posterior and lateral on the testis. It does have three regions: a head, a body, and a tail region. And there are some slightly different things that occur within those different areas, but we're not going to emphasize that and worry about too much what's going on. Instead, we'll function on the three major functions that occur in the entire epididymis. Those three functions include monitoring and adjusting the composition of the fluid, making sure that there are correct nutrients and things like that to provide the nutrients and maintain the sperm within the epididymis. And again, remember, they're going to be in there for about 20 days. Here in the epididymis, we're going to find a unique tissue that we do not see anywhere else in the body. And we'll actually see the same thing in a female as well, different tissue. In this case, what we have here is a ciliated pseudostratified columnar epithelial tissue, which still just rolls off the tongue and that you enjoy immensely. However, what's different about this ciliated pseudostratified columnar epithelial tissue than any other ciliated pseudostratified columnar epithelial tissue you run into in the body is that here, the cilia are non-motile. We have cilia, but they do not move in that wave-like motion. Again, it seems like it would make all the sense in the world. Having motile cilia is what moves mucus in our respiratory system. And since we don't want the sperm to swim, why not have these motile? And I don't have a good answer for you. Instead, they're just there to increase surface area for the diffusion and absorption of materials to maintain the sperm. So why they don't move, I don't have a good reason for you. Maybe they would move too fast to move the sperm through it too quickly. So maybe it's not advantageous that way because they need to be in here for 20 days to mature. And of course, that's what they're going to do. They're going to store and mature the sperm for about 20 days. But not again, anytime you're making 500 million of something a day, right, there's going to be some errors in that process. And so the other important function of the epididymis is to be able to recycle damaged or abnormal spermatozoa. You might not have thought of it in these terms, but this is probably something you're aware of if you're someone who's ever had difficulty getting pregnant. If you and your significant other are trying to get pregnant and having challenges, when you go to the doctor doesn't encourage you to have sex 14 times a day and 17 on Sundays, right? As much as you possibly can, right? Flood the area with semen and hope for the best. No, you typically wait outside of the point where she is ovulating. And even when she's ovulating, you only have sex 1 or 2 times every other day around that time, because it gives, if you release sperm too quickly, then obviously sperm production has to increase. And the faster you're producing the sperm, the less time there is for maturation, the less time there is to filter out dead and abnormal spermatozoa. And so the overall viability of the semen actually goes down. So there is an advantage to this, to have taking the time for it to be able to go through this process properly and be able to mature and be recycled so that the sperm that are produced are going to be more viable. After about 20 days, they then leave the epididymis and leave the testis and scrotum back into the abdominal pelvic cavity via what is known as the Ductus Deferens, or also known as the vas deferens. And both terms are acceptable terms. That Ductus Deferens takes the sperm back into the abdominal pelvic cavity, and it goes, as we talked about, through that inguinal canal, through the spermatochord. Once again, if you are gentle, one of the things that you can see here from this illustration is that the Ductus Deferens actually comes up and over the pubic bone before it enters into the inguinal canal, into the abdominal pelvic cavity. So if you gently palpate the pubic symphysis of the male, start at the pubic symphysis and move slightly lateral, you can actually palpate and feel that tubule as it is entering into. Again, be gentle, be kind, but you can actually palpate that ductus deferens, the vas deferens, as it is carrying the sperm into the abdominal pelvic cavity. Notice it takes it up and over the bladder, around the back, and actually gets hooked by the ureter coming down from the kidneys, where ultimately it then feeds into the ejaculatory duct, where it meets up with the secretions of the seminal vesicle and are released into the urethra. But we'll get there in just a second. So again, the ductus deferens' job is to carry the sperm through the inguinal canal into the pelvic cavity, over the pubic bone, as we talked about, where, despite what your male significant other tells you, the sperm can be stored in the ductus deferens for months, right? It does not become toxic if it stays in there for more than a couple days, so it does not have to be released to save that loved one's life, regardless of what they're pleading with you on. It can stay in there for months from that process. And again, it is going to be moved through the ductus deferens out into the urethra via peristalsis. Again, we don't want those sperm swimming until they're on their way to the vaginal canal. Of course, the male form of birth control is that vasectomy, and with that vasectomy, we have a cutting of the ductus deferens, and I think we'll see this best if we go back to the previous picture. One of the advantages of this process is that it is a simple surgery that can be done as an outpatient surgery. It is often done as a weekend surgery where you have it done on Friday. You spend the weekend with a bag of frozen peas on your testicles, and Monday you're back at work again. Back in ancient times, and we just passed the famous time for this, right? March Madness is one of those times where males are wanting to sit on the couch and watch basketball for hours upon hours on end. And several years ago, there was a urologist, and I believe it was Wisconsin or Minnesota, someplace like that, that actually did a vasectomy special. Basically, he would do the vasectomy on Wednesday, so that you had then could sit. You were forced to sit with the ice on your testicles for the next 2 or 3 days. So Thursday, Friday, Saturday, and Sunday's games, and he actually threw in a free pizza as well. So you get a vasectomy, get a pizza, and get to watch basketball, all medically. And not surprisingly, he had a tremendous number, over 1,000 different people who wanted to sign up to do this as a result of it. Again, it is much safer, much easier surgery than it is for the female, where we have to go into the abdominal pelvic cavity to tie her tubes. One of the interesting things about this process is originally, when it was first done, basically all they would do is go in, that's not going to be big enough. They would go in and put a clip on the ductus deferens. The advantage of a clip is that it could potentially be reversible, right? Let's say something horrible happens to your wife. She's in a horrible accident and is killed, or maybe because of she's staying at home with the coronavirus, she's put on 20 pounds or whatever it is, and you need to get rid of her. So as a result of that, you want to have a new wife and new babies, right? That clip could be removed, right? However, what they found is those clips weren't very sturdy. So they often would slip off and you would get a bunch of surprise pregnancies. So they stopped doing that. What they did next was that they would just go in and do an incision. They would just cut it. But again, what they found is that on occasion, the two cut ends would actually grow back together. So now what they do is they actually cut out a section of the Ductus Deferens and they cauterize the ends of it, basically making it close to zero. There are still some very rare instances where something really weird can happen. But in this case, it is a completely, and again, they consider it irreversible procedure. I joke about, you know, your wife gaining weight or things along those lines, but prior, because again, most of these are done by HMOs now, prior to getting the vasectomy done, you were actually required to go and take a psychological course where they actually talk to you about, all right, what happens if your kid ends up having one of those horrible genetic disorders where having another offspring that could give them a kidney is the only way they can survive? Or what if your wife and kids are killed in a horrible accident, right? You are not going to be able to have kids anymore. So they encourage you to either store and house your semen, get in a sperm bank or something along those lines, or they want you to consider it a one-way procedure. And again, it is a very benign, very simple procedure. In some rare instances, you may get abnormal swelling and pain. And in those cases, it can sometimes be resolved by actually reconnecting the ductus deferens, and so they will do that surgically there. However, without that increased swelling and tremendous pain, technically it might be possible to reconnect them, but most HMOs won't cover it, so it's something you have to pay for out of pocket. And here's the other problem. Again, they make the cut. It isn't just about changing the piping. We cut the pipe, we take the piece out, we cauterize the end. So even if you could connect them together without the scar tissue, what actually happens in the testis is that for the first few months, there will be continued sperm production. But eventually, without the ability to release the sperm production decreases dramatically. So 2 years later, if you try to get your vasectomy reversed, it's quite possible at that point that the testes will not be producing enough sperm, or even if you were able to reconnect the tubing, you wouldn't be able to produce enough sperm to be able to viably impregnate an individual. So again, but like I said, it is much, much better, much, much safer than the female version of this, the tube tying, which we'll talk about when we get to the female system. All right, questions on that? Excellent. So as we talked about, let's finish the rest of the ductwork system. Now notice we are looking at a posterior view of the bladder. We should be able to tell that because here we see the ureters coming down, because we know the ureters also enter the bladder at the posterior and inferior side. Our ductus deferens comes up and hooks over the ureter. Notice then there is an enlarged region known as the ampulla, which stores large amounts of semen ready to be released upon ejaculation. This is also where those seminal vesicles, those seminal glands, are located. And if you notice, the duct of the seminiferous vesicle and the ampulla of the ductus deferens come together to form a small duct structure that penetrates through the prostate into the urethra, and that is known as the ejaculatory duct. And so the ejaculatory duct goes from the ductus deferens into the urethra. And then, as we already know, the male urethra has three parts: the prostatic urethra, the membranous urethra, and the spongy urethra. And from there, you are out of the male reproductive system. These ampulla are important because of two reasons. They, again, are large swellings that can contain large number of sperm in them. They're going to meet up with the seminal vesicles to form the ejaculatory duct, and they enter into the urethra. So it keeps a large amount of sperm close by so that when ejaculation occurs, powerful peristaltic contractions can release a large amount of sperm into the urethra to help to form the semen. However, these ampulas become active upon arousal. When a male becomes aroused, they will start to contract these ampulla, which can release some sperm into the urethra prematurely. This is one of the reasons why withdrawal is not a viable method of birth control, because any penetration of the female has the potential, because if the male is aroused, there can be sperm in the fluids that are being released from the male, even if he hasn't ejaculated yet. In fact, the concentration of sperm, because it's such a smaller volume of fluids that are released in this process, the concentration of sperm can actually be higher in the pre-ejaculate than actually in the ejaculate itself. And so even if you withdraw before ejaculation, there is plenty of opportunity for the presentation of sperm to the vaginal canal. So again, withdrawal is not a viable method of birth control. As we talked about, the urethra has the 3 parts. It extends from the base of the bladder through the penis. Prosthetic urethra passes through the urethra. Membranous urethra passes through the levator ani muscle or the urogenital diaphragm. And the spongy or penile or cavernous urethra passes through the penis. So again, a completely closed system. Seminiferous tubule, straight tubule, rete testis, efferent ductule, epididymis, ductus deferens, ejaculatory duct, prosthetic urethra, membranous urethra, penile urethra, out. Hey, Macarena. All righty. And that is the mystical, magical journey that our sperm make. However, as we've kind of not just hinted at, but blatantly said, semen is not just sperm. In fact, the sperm and the fluids that are produced by the nurse cells and by the epididymis only make up about 5% of the fluid volume of our semen. So it is those 3 accessory glands, the seminiferous, the seminal vesicles, the prostate, and the bulbourethral glands that produce the majority of our semen. Also, if you look at that numbers, because one of the questions that is often asked is that if a male has a vasectomy, does it decrease the volume of the ejaculate? And as you can see here, while the answer might be yes, not by very much. If the testis and the epididymis are only providing 5% of the fluid, you're still 95 being produced by those other glands. And so in most cases, you do not see a noticeable decrease in the volume of ejaculate produced if that is something that is important to you. Now, aside from making up the bulk of the semen, obviously that must be produced. Produced for an important reason, and there are really a couple. As we said, up to this point in time when it is being released in the ejaculate, we do not want our sperm wasting their energy, wasting their metabolic resources, spinning their tails. So the first thing we need to do is activate those spermatozoa, get that tail moving. That tail motion requires a tremendous amount of metabolic energy, so we need to be able to provide the nutrients necessary to maintain that motility. We also need to stimulate peristalsis, not just in the male for the expulsion of the ejaculate, but also in the female's reproductive tract as well. One of my absolutely favorite things to say in this class is that the female's reproductive tract is a hostile environment. Right? It's very entertaining to say that, but it's also vitally important and true. As I mentioned previously, the female's reproductive tract is open to the abdominal pelvic cavity. So anything in a harmful bacteria or other materials in the outside world have the ability to not just get into the reproductive system, but to get into the abdominal pelvic cavity and be able to affect any of the organs in the abdominal pelvic cavity. So yes, the vaginal canal is a hostile environment, but that is what we want it to be. It needs to be that to protect that female. It's vitally important that it be that way. But that hostile environment's not good for sperm. So we need to help to neutralize some of that acidity that's in there, neutralize some of the hostility. And if we can stimulate peristalsis in the female's reproductive tract, that can help to facilitate the movement of the sperm through the vaginal canal, through the uterus, up to the uterine tubes where fertilization will hopefully take place. So again, not only do we need to stimulate peristalsis in the female, but we need to protect the sperm and help to neutralize the acidity because the vaginal canal is a hostile environment. All right, I think I'm gonna have to put that kids warning on the lecture for this one when we're presenting this. Not that kids are randomly searching YouTube to find out about lectures of anatomy and physiology, but all right, excellent. Let's talk a little bit about these glands. Your book does a great job of talking about all of the components and all the composition. And again, in the interest of time, I don't want to spend too much on that, but we do need to talk a little bit about what's going on. Starting first with the seminal vesicles. Again, they are paired. There are two of them. They're the well, not the largest. The prostate is the largest of these, but they're relatively large glands located on the posterior inferior part of the bladder. While not the largest gland, they do produce the largest amount of the semen. They produce a thick, yellowish, alkaline secretion that makes up about 70% of the semen. This contains a lot of important components in it. Fructose, again, to give it that sweet flavor. No, actually to provide the energy necessary, that sugar necessary for the production of ATP. Ascorbic acid, what is that again? That sounds vaguely familiar. What's the other name for ascorbic acid? I know you're all asleep because I haven't asked too many questions because I know it's awkward to talk about these things on here. But someone, say again?
Vitamin C.
Vitamin C, there you go, because we don't want those sperm getting a cold. No, actually, the vitamin C is there actually to help to. It's one of the components that helps to activate the sperm and actually start the swimming motion. Our seminal fluid has a coagulating enzyme because, again, we want the semen to stay where we put it. Right, the goal is to place the semen into the vaginal canal and have it stay in the vaginal canal. So by having a coagulating enzyme, it helps to maintain more of the fluid inside the vaginal canal. So that increases the likelihood that the sperm will get to its destination and its goal of fertilizing that egg. One of the most important components of the seminal fluid is a hormone called prostaglandins. Prostaglandins are actually a class of hormones, and what they do is they help to trigger peristalsis. In particular, they help to trigger peristalsis in the female. Normally, the female's reproductive system has a minor peristalsis outward from the uterine tubes through the urethra out the vaginal canal, right? We don't want anything that's in there coming up into the abdominal pelvic cavity, so normal peristalsis would be outward. But what these prostaglandins do is they can produce a reverse peristalsis. And that reverse peristalsis can help to draw the sperm up towards the uterine tube for fertilization. This is significant for a couple of reasons. One, again, it helps in fertilization, but the other concern is this is one of the things, and again, not everybody's necessarily thought of it in these terms, but one of the things that they do warn pregnant couples about is having sex late term in the pregnancy. And the reason for that is twofold. One, the female orgasm, with the female orgasms, that can cause vaginal contractions, and those contractions can stimulate the positive feedback process that could lead to early parturition or birthing of the child. But these prostaglandins can also cause that reverse peristalsis could also trigger the onset of uterine contractions as well. And again, it's also one of those old wives' tale, one of the things that they talk about if you're late term, you know, 41, 42, 43, and still haven't given birth, one of those old wives' tale is what got you into the mess can get you out of the mess. And that's exactly what they're talking about. Intercourse at that late stage, week 41, 42, whether it's the female orgasm or whether it is the prostaglandins, can trigger contractions, and those contractions can trigger the uterine contractions that can lead to that positive feedback that can lead to expulsion. All right. There. There are lots of other substances and other components to the seminal fluids. And their job is not just to turn on the swimming motion of the sperm, but also to allow the sperm to be able to fertilize the egg. Think of it this way. If I instead had a petri dish, and I had an egg sitting in that petri dish, and I was able to, with a needle, come into this ampulla of the Ductus Deferens and pluck out a sperm and take that sperm and put it directly on the egg, nothing would happen. Even if we removed all the egg's defenses, that sperm would not be able to fertilize the egg. Because not only does it have to start the swimming motion, but it has to be able to prepare to become able to fertilize it. Again, think of it in terms of our immune cells. We had immune cells that were mature, but were naive, right? They were capable of an immune response, but they hadn't been exposed to the right pathogen yet. So they weren't dividing, they weren't making antibodies, they weren't killing other cells or doing any of the other things those immune cells were. They were ready to do it, but they weren't doing it yet till triggered. Well, that's what happens here. Here in the Ductus Deferens, we have mature cells, mature sperm that are ready to, are capable of, but aren't ready yet. And that process of getting ready to fertilize the egg is a process we call capacitation. That process of capacitation can actually take hours, right? And again, we always on the TVs and the commercials, on the videos, on the cartoons, it's always the first one that gets there, but it's not about the first one that gets there. While it may only take a few minutes for a sperm to get to the egg, it still may be hours before it actually is starting to be capable of fertilizing that egg, because it takes time for this process of capacitation to become able to fertilize the egg. And so that's an important process that the chemicals of the semen allow for. The next gland is the prostate gland. It is a donut-shaped gland that sits at the base of the bladder, right? All males have it. And again, one of the things that happens with age is that the prostate enlarges. As it enlarges, it can restrict the prostate. That's why back in ancient times when we had sporting events, it was impossible to watch a sporting event without seeing a handful of prostate enlargement commercials, right? Because again, you get that irritation of the urethra, that sensation you need to avoid, or a restriction of the flow because of that enlargement of the prostate. It is a donut shape and it completely encircles the urethra. Typically, the illustrations always look like this, where you have these striations to it, because it has many, many, many small ducts that all secrete out into the prostatic urethra. So that's why it always kind of appears to have this striped appearance to it, because of those many, many ducts that release into the prostatic urethra. It produces about 30% of the semen. Again, notice these numbers don't equal up to 100 because we're rounding on some of these things, because the exact number can vary and isn't important. And again, it has dozens of different special chemicals in there that primarily play a role in activating the swimming action of the sperm and capacitating the sperm so that it is ready to fertilize the egg as well. But like I said, also notice between the semen, between the prostate, and between the seminal vesicles, we're already over 100%. So we haven't yet talked about the bulbourethral glands, and we're already at 100% of this seminal fluid, over it really. And we'll see why that is in just a minute. There is one more thing I want to point out about the prostate. Yes, prostate enlargement is a common problem that occurs to most males as they age. But prostate cancer is another thing that happens to many, many males as they age. And while it has the potential to be a deadly cancer, it does tend to be one of the slower growing cancers in most situations, but it's also highly treatable. So what is done to a male, typically once he reaches the age of 50, if you think of, here we see the sagittal anatomy of this, the prostate is located right here next to the rectum. So that male on his 50th birthday or shortly thereafter gets the pleasure and enjoyment of having their doctor insert a finger into their rectum so they can palpate the surface of the prostate. What they're looking for is twofold. Both they're looking at the overall size of the prostate, but the other thing they're looking for is any irregularity on the surface that could indicate tumors of the prostate as well. Of course, you don't have to be 50 to get a finger inserted into your rectum and have your prostate palpated. There are some individuals, no judgment, this is a judgment-free zone. Your prostate is an area that contains sensory organs, sensory receptors. And so for some males, the stimulation of the prostate, that palpating of the prostate, can be seen as an incredibly pleasurable sensation that can enhance orgasm. So there are some males that appreciate that, and we'll make a point of emphasizing that because we see something similar in females as well. Alrighty. What that leaves us with, as I mentioned, are these last two glands that are the bulbourethral gland, or also known as the Cowper gland. Cowper is an appropriate anatomical term. You know, good old Bob Cowper is the first person who identified them. So if you want to call them Cowper glands because that is easier to spell, that is fine. I will accept that. But bulbourethral is what they're commonly known as. There are a couple interesting things about them. They're paired glands. They are small, about the size of a pea. And notice the gland themselves are embedded within the urogenital diaphragm, within the levator ani muscle. Within the skeletal muscle is where they are embedded. And remember, the part of the urethra that is surrounded by the urogenital diaphragm is the membranous urethra. But if you notice, while the glands themselves are located in the urogenital diaphragm, their ducts don't actually express into the membranous urethra. Instead, notice their ducts come forward and express into the spongy urethra. Remember, here in the muscle is where our external urethral sphincter would be located. So notice technically your external urethral sphincter can be constricted, but you can still release secretions from the bulbourethral glands. And these bulbourethral glands do produce secretions. However, if you notice, they are not a major component of the semen. And the reason for this is that the bulbourethral glands become active upon arousal. So these are not, don't become the prostate, the seminal vesicles, they produce and release their secretion upon orgasm. But the bulbourethral glands release their secretion upon arousal. They produce a thick, clear, alkaline mucus that is a pre-ejaculate. This is that pre-ejaculate that, as I mentioned, can have sperm from the peristaltic contractions of the ampullas in them. And this pre-ejaculate is typically a clear, thick, alkaline mucus that serves a couple of functions, right? One is, again, remember in the male, this urethra is also a passageway for urine. And one of the things that we mentioned is that acid is bad for the sperm, but we really haven't said why. One of the main reasons that acid is bad for sperm is in an acidic environment, the sperm swim more slowly. So not only can the acid be damaging, it decreases the motility of the sperm as well. As we just learned, urine is primarily acidic. So cleansing the urethra with an alkaline secretion can help to protect the sperm. The other thing about this mucus secretion is, again, it is lubricating to help to facilitate copulation, to make copulation easier. Both the male and the female release alkaline mucousy secretions upon arousal to facilitate copulation. And again, being alkaline, it can help to reduce the acidity of that hostile environment that is the vaginal canal. So basically, it helps to clean the pipes a little bit, helps to facilitate the copulation, and then helps to hopefully make it a less hostile environment for the sperm, making them more likely, hopefully, to be able to fertilize the egg. All right, we put it all together, and we get that magical, mystical combo that we call semen. It is, again, a mixture of both the sperm and the secretions from the accessory glands. As we talked about, it is primarily basic, typically is milky white in appearance. Again, a typical male ejaculate is typically about 2 to 5 milliliters of semen. And like I said, only about 5 to at most 10% of that is sperm. The rest of that is the combined secretions of the glands. But even though the overall volume of sperm is relatively low, the number of sperm is incredibly remarkable. A single typical ejaculate can have somewhere between 20 and 150 million sperm per milliliter. And again, if the ejaculate has 5 milliliters, to keep the, we'll go 4 because the numbers are easier for that. With 4, that would be 600 million sperm, right? With 5 milliliters, that could be almost 250 million sperm in a single ejaculate. That is a massive amount of sperm to be released. And again, why do we need so many? Well, mostly because none of them will stop to ask for directions. No, because there's a massive attrition in sperm. That sounds like a large number, but if you have less than 25 million per milliliter of ejaculate, you are considered to have a low sperm count. And if it is below 20 million per milliliter, you are considered infertile. You are considered incapable of getting a female pregnant. All right? Again, 20 million per milliliter is a massive number of sperm, crazily big number of sperm, but it isn't enough because of the massive amount of attrition that takes place between getting from the vaginal canal to the uterus, from the uterus to the uterine canal, and then to break down those defenses of the egg. Basically, you're infertile if you have less than 20 million per milliliter. Again, these numbers are astronomical. If you think about how many sperm a male produces and releases during the course of their life versus how many eggs a female produces and releases during the course of its life, their life, the numbers are staggeringly different. All right. And like I said, typically at least 60% of the sperm are fully functional and viable. But like I said, with repeated ejaculation, that number can decrease dramatically. So like I said, when the goal is to get someone pregnant, the goal isn't to have sex as frequently as possible, because the more sex that's had, the less filtering of the sperm takes place, the less volume of sperm you have in relation to the ejaculate. And so the overall count goes down and the number of viable sperm go down as well. And there you go. With that, we have made sperm, we have mixed it with secretions, and we have released it from the male body. With that, that is everything that I wanted to talk about for today. We still have a little bit more anatomy to talk about in the males, and then the physiology of the males. And the physiology of the males is going to take about 3.5 minutes. Okay, not quite that, but it is very simple and straightforward. It is very basic. So again, we probably won't have to spend more than an hour on males on Wednesday, and then we'll get to spend the rest of Wednesday and all of Monday talking about females. All right. Again, because of the transition and all the craziness that happened, we did lose a couple days. So what's likely going to happen is that we are going to probably lose some of the end of this when we get to talk about, normally we talk about genetics, normally we talk about development. I don't know how much time we'll have for that. We'll have to see what next lecture looks like. And I'll have a better idea on Wednesday, at the end of Wednesday, what we're going to be covering on Monday. Because I don't want too much new information 2 days before the exam, especially because that tends to be some of the more challenging stuff with the more challenging vocabulary. Again, you could spend literally a whole semester talking about development and heredity, and there are classes on that. Plus, there's all sorts of fun for terms like syncytiotrophoblasts, which you have to spell and all those things. So we'll see how far we get on Wednesday and decide what's going to happen on Monday. All righty. Also, after going through this, I think it will be beneficial. So I am going to try to set up and be ready for a histology review on Friday. So I'll try to work on that between today and Wednesday so that we can have that set up for Friday as well. Because I think from most of the feedback that I got, that worked well for people. Also, please take that survey. It's under the quizzes. About the lab exams so I can see what you guys liked better and what worked better for you guys for that. All right. Any questions on any of that? All right. With that, we are done with all the lectures. As always, have a good day. Be safe. Study hard. And I will see you guys on Wednesday. All right. I will stop the recording now.